Hi, this is Dr. Jabali, and in this video we're going to do plant growth, which is topic 9.3, which is all about auxins and meristems. So, what are the things that we're going to target on? We're going to talk about undifferentiated cells such as meristems that are found in plants. We're going to talk about how the shoot apex has to do with extending the stem by mitosis and cell divisions, basically of undifferentiated cells such as the meristems. We also going to talk about how plant hormones can control the shoot apex, which means the growth of the plant. We're also going to talk how the shoot responds to the environment by tropism, which that means depending on the gravity, depends on the light, it will tell you which way the plant will grow. We're going to talk about auxin, which is a hormone that has very much to do with the way in which the plant is going to grow. We're going to talk about how it is influenced by the parents and changing the parents of the gene expression. We're also going to talk about micropropagation in plants, which you can actually create new plants by using nutrient agar gels and hormones. We're also going to talk about how using micropropagation right here, we can do many other plants that are virus-free from strain of existing varieties, and of course, continue with the propagation of orchids and other different rare species. So, what we want to go to here is talk about the meristem. When we say the meristem here, just seeing the word meri right here and the word stem, it's obviously that we're talking about stem cells. But it's stem cells in what? In plants. Now, where is the location that you can find these meristems in plants? So right here, we have a plant with, of course, the soil, and we have the tip of the plant, and we have the tip of the root right here. So once again, I put here soil for you to see. And then here you have some buds on the sides, right? Now, these basically the locations of meristem is these ones. So you have basically at the tip of the root right here, you have at the tip of the stem, and you have on the buds on the lateral side. Oops, let me write here better. Lateral sides. So all of these little black spots that I show you are places where you can actually find the stem cells of the plants. That's why the plant can grow in this direction, down this direction, or sideways. Just to show you, if there's more roots, of course, if the roots are like this, then all of them will have the tips. So once again, these are where you can find the stem cells. So we're going to actually look more details, what's their names, and why some of them grow at a certain time and some grows in other times. So plant tissues, meristematic tissues, these are source of new growth for the entire life of plants, right? There's the primary growth and the secondary growth. When I say primary growth, that means they're going taller or deeper. As you can see, they're getting taller and deeper like this. So these only happens at the tip of the roots and the shoots, and that's what I was talking about here, these parts right here. These are known as the primary growth. So I will put here as a primary growth. That means going up or going deeper down this way. Now, the second one, as you can see here, it is the plant grows wider, so you can see it sideways, and known as the vascular or the cord cambium, which is the lateral meristems. So these ones right here, these are known as the lateral meristems. Now, this is known as apical, if you want to call it. The tip one is the apical meristem. So apical for the primary growth, lateral meristem for the secondary growth. And of course, these here is called the secondary growth, which is basically going sideways. So you can see the difference between the two types of growth that happens in plants. We have the primary growth, which is going up or down, and the secondary growth, which is going sideways, so making more the thickness of the cambium. So these are the two ways in which the plant grows, and all of them have the stem cells. So here's more focus on looking at the root. So we have three sections of basically of the root, right? We have the zone of division, the zone of elongation, and zone of maturation. See, here is basically the apical meristem is located on the root cap, which this means this is where most of the stem cells is really actually located. And that's usually a place where you find the undifferentiated cells, and this is usually when they're just starting to get to make many of those stem cells. The zone of elongation here is where the cells are enlarged and they are starting to get to G1 phase, so they're starting to get a certain function around here. And zone of maturation, these guys have finally figured out what their function, and that's it. They have figured out what is their job for the rest of their life. So these three zones are in different stages of the cells of the plants. But once again, where you find the stem cells, right here at the zone of the cell division at the apical meristem. Now, next thing is talk about is the hormone auxin. Now, auxin in the Greek world means I grow or increase. And they were basically one of the major plant hormones to be discovered. So auxin is the biggest one here, known by the Greeks both the times. Now, what is the auxin hormone used for? Of course, we say cell growth and division on the meristematic tissues. That means the growth and cell divisions of the stem cells. They also help differentiate the xylem and the phloem. They also promote axial elongation on the shoots and lateral expansion on the root swelling. They stimulate the growth of flowers and the induction of fruit production without pollination. These are just giving you some ideas that you want to do your IAs on, but these right here are just samples of what the auxin can be done with auxin, but the one that you must really know is definitely the cell growth division of the meristems tissue. This is the one that IB expect out of you when it comes to auxin. So the next thing is how does auxin is related to phototropism. So first, let's understand what the word tropism means. And tropism means the growth or the movement to a directional external stimuli. So that means something in the environment is causing a plant to move. That means it can be the gravity, it can be the light, it can be the temperature, anything out there is causing the plant to move. That's called tropism. Now, when it's moving towards, it's positive. When it's moving away, it's negative. Okay, so if it's moving away from you, we consider it as negative. Away will be like a root going downwards. Or if you're moving towards you, you will see a plant moving towards your direction. Now, what are the examples that can be these external stimuli? They can be chemicals, gravity, touch, light. These are just examples of stimuli here that can cause the plant to move away or towards you. Okay? So phototropism here, from the word photo, you can think of the word light. So that means light is causing some kind of growth. So when this happens, again, auxin has a very important part in this. So in the shoots, right, high concentration of auxin promotes cell elongation, meaning that dark side of the shoot elongates and the shoot grows towards the light. So again, the word photo here, it means light. It's a light that is pushing the growth of the plant. So this part basically means if the shoot grows towards the light, that's a positive phototropism, right? Positive, that means it's going towards the light. The lower side of the shoot elongates and the root grows away from the ground. So this is the case when it comes to shoots. Now, when I say the shoots, I'm talking about the stems, okay? So in another sense, and the dark side of the stem elongates while the stem grows towards the light, and the lower side of the shoot, right, which is in the bottom part, it elongates and the root grows away from the ground. So this is very important that you remember these guys because you need to know the difference between the shoot, which and the next one is about the roots. High auxin concentration inhibits cell elongation. Look at this. Roots inhibit, the auxin inhibits cell elongation, meaning that the dark side root becomes short and the root grows away from the light, which is negative phototropism. The lower side of the root becomes shorter and the roots turn downwards into the earth. So what you can understand from this, if I had to highlight and reinforce the information here from both, it's just basically what it's in red and in bold. So when you talk about the stems, and it's all about high auxin, okay? In the stems, it promotes cell elongation. And that means the shoots or the stem grows towards the light. And the lower side of the shoot elongates, okay? When it comes to the root, we can say that they stop cell elongation. And that means the roots grow away from the light. So when there's a high amount of auxin, the roots will move away from the light, which is basically negative phototropism. And the lower side of the root becomes shorter, the lower side, okay? And the roots turns downwards into the earth. So this is something that might come out as multiple choice. So what we have here is an exposure of the sunlight, and these little pink things are auxin, as you can see here. So when the sun is in exposure right here in the day, you can see that the auxins are kind of spread around. But then afterwards, you can see that the sun is here, and look where the auxins are going. They're going on the other side of the sun. And that leads eventually to the bending to happen. Why? Because the auxin always move opposite of where the sun is located. So, and when this happens, auxin on this side will cause some kind of different conformation of the cell plant and will lead it to stretch. And this is what happens. You kind of stretch. But what it do? It eventually made the plant move towards the light. But this all started with one thing and only. If the sun is on this side, the auxins will literally go to the opposite side. So let me show you this more understanding on the plant. So let's say you have the sun on this side here, and you have the plants. If the plants are here, the auxins, in this case, they all move to the outside. That means that the light is hitting on this side right here, but the auxins, they move to the opposite side. When this happens, this will cause a change in this area. And eventually will cause what? It will cause the plant to start doing this. And it only did this because the auxin somehow changed, like I told you, this area of the plant, so it made it stretch more. So now it bends towards the sun. Now I can do this the other way so you can see what I'm talking about. Let's say the sun is completely on the opposite side. The sun is here, and you have the plant. Again, auxin will move to the opposite side of the sun. And when it moves to the opposite side of the sun, it will cause a conformation in this area. So this is what it's going to look. It's going to look like it's bending this way because the oxygen is causing a change in this area, and it moved towards the sun. So this is important that you need to know which way auxin is moving according to where the sun is located. Next thing we're going to do is learn what exactly does the auxin do inside of the stuff. So before we get to that, first let's understand about apical dominance and pruning. So apical dominance here, it just means that we actually, right here, highlighting here, the terminal bud releases auxin, right? Inhibiting the growth of the lateral buds. That means if you're going to increase just this part, this area, right? What happens? You are basically stopping the growth on the sides. But if you do the opposite direction, if you decided to cut the apical bud, right? You're destroying the apical bud, and this way only allow the growths on the sides, then you're going to see that there is going to be growth of branches. So for example, by pruning, that means if you cut the apical bud, the only thing you will have is the growth of the branches on the sides. Once again, if there is a growth happening here, not much growth is going to happen on the branches on the sides. But if you cut this growth on the top, then you're going to see all the cuts coming in. So removing the terminal bud via selective pruning allows dormant lateral buds to develop, the producing busher plants. That's what it means. So here I'm going to show you an example so you can understand what I mean. So you have a plant here. Actually, no, let's make it smaller. Let's say if it's tiny like this, and you have all here, this area, what do you have? You have the stem cells, right? So if you allow this to continue life, what's going to happen? You're going to continue growing. And of course, the stem cells continue to grow, and we continue to grow. That is if you still have the stem cells at the apical bud, which is this one. Now, what happens to the sides buds, right? The lateral ones. They continue to be shut off. That's what happens. They continue to be shut off. However, if I decided, let's put it here, cancel. If I decided to cut this one right here, I cut the apical bud. It's been cut. Now, what happens with this? This doesn't happen, this doesn't happen. But what we start seeing is something completely different that's going to happen here. And what is exactly what you're going to see here? You're going to see branches going through the sides, right? So you're going to see, and then there's going to be more branches, more branches, more branches, more branches, more branches going to the side. So you're going to allow the lateral buds here to start growing. So this is what pruning means. You're just basically cutting the apical bud to allow the lateral ones, lateral buds to start moving. That's what it all means. This is what the whole pruning is. Now, when I say apical dominance, let me explain what apical dominance from pruning. Apical dominance means it's just that it dominates. So it's what I drew the first time. So if you do have the apical dominance right here, it, because of what's happening in the growth, if the growth is happening here, it basically inhibits the growth on the sides. It won't happen. As long as there's growing here, or even the roots, if there's growing roots here, the side ones are inhibited. It will not happen. Now let's understand here how that growth happens on the sides when we talk about the whole movement here of the auxin. So it all starts with the auxin stimulating the hydrogen pump and causing the hydrogen in the cytoplasm to move towards the cell wall. In the cell wall, we have cellulose. So what does the hydrogen do? It actually activates the enzyme expansin, which basically loosen up the cellulose. So look how they were tied up here, the cellulose, and look how they became a little bit more loosened. That leads to an elongation of the cell and allow, you see the vacuole here, how small it is, and then it becomes bigger. So why? So by adding water to the vacuole, now the cell, look, it got way bigger than what it was before. So the mechanism of auxin is just simply to break down the cell wall, and by breaking the cell wall, you can stretch the cell even bigger. So here, more specific, look at this. You have basically auxin and activating the hydrogen pump. So these hydrogen cytoplasm move in. And what do? They activate the enzyme, which eventually does what? It starts breaking down the cellulose. So the enzymes come and start breaking the cellulose on the wall. And now because it broke them up, what happens? The water inside the vacuole start causing bigger pressure. And look, you were a tiny cell to become a bigger cell just because the water went into the vacuole. Now, a lot of students say, oh, once you loosen up the cellulose, right, with the enzyme, it's done. That's not enough. It's not just about the enzyme breaking the cellulose in the cell wall, then it elongates. It also needs to put the water inside the vacuole and cause the turgor pressure. That's the whole explanation of auxin. So when it comes to auxins and cell elongations, the sequence of events in the stem causing it to bend towards the light source is as follows. So here is a full written sentence of how the auxin does. So starting, auxin is produced by all cells in the stem on the side towards the light source. Auxin moves by efflux pump action into the nuclei of the cells on the side of the stem opposite the light. So remember, auxin will always move to the opposite side. Auxin receptor in the nuclei form a complex that activates the proton, the hydrogen pump. The present research indicates that auxin itself also has an effect on the protein pump at the stem cell membrane indicated. And forget about the picture here, but it's just basically saying that auxin is the one who activates the hydrogen pump. The proton pumps moves hydrogen ions into the space of the cell wall. The hydrogen ions cause a drop in the pH, resulting in the hydrogen bonds between the cellulose fibrous cells to breaking. Now, this is more detail how the enzyme and the hydrogen does that. As a result, the elongation of the cells on the side of the way of the light. Now, here they're not continuing the whole thing, but I'm going to show this through a drawing here. Let me actually add a drawing here so you can see what I'm talking about. So how does this process all starts? It starts with auxin, right? Now, auxin moves to the place away. It moves away from the sunlight. So basically in a plant cell, right here, you can see the auxin will be on the side that is away from the sun. So it's packing on this side. So when it's packing on this side, let's zoom in more closely. You have the cell wall here, right? This is the cell wall. And what are these lines here? These are the tight cellulose. Because if you remember, cellulose, what does it do? It makes up the cell wall. So right here, we have auxin. I'm going to put here, auxin again. What's the first thing it touches? It touches the hydrogen channel. So this hydrogen channel, and I should say actually pump. Let's say pump here. So the hydrogens that are found inside of the plant cell in the cytoplasm will move towards the inside, and then there is an enzyme. In this case, the enzyme is expansin. And what does this expansin do? It loosens up the cell wall by breaking cellulose. And if you're breaking cellulose, now this whole area here, it became loosened up. Now that's not enough. Nope. We have basically inside here a vacuole. And this, once the water goes in here, that water will cause the vacuole to get even more bigger like this. And then that's going to even stretch more the cell to become bigger as it was before by doing this. It's just stretching the entire vacuole. Oops, let's say it was supposed to be in blue. So you can see the vacuole became bigger and bigger just because the cellulose here has loosened up. So now what happened? You kind of stretch the whole thing. So in this process here, it all starts, and we're going to write this down very quickly so you can know for the test, is 1, auxin moves away from the sun on the opposite side of the plant cell. Number 2, auxin activate the hydrogen pump, so the hydrogen move from the cytoplasm into the cell wall. And then number 3, the hydrogen activate the enzyme to break cellulose. Number 4, the cell wall becomes, the word loose, becomes loose. Number 5, water enter the vacuole and the cell elongates on the side where auxin worked. So this will be showing what will be like a plant moving and elongating on this side as basically the sun is hitting the plant. So you can see the motion going. So this is basically a very straightforward mention of the points. First, you got to say the auxin is moving away from the sun, which is opposite side of the plant. Auxin activate the H pumps. You're going to mention the hydrogen moves from the cytoplasm into the cell wall. You're going to say the hydrogen activates the enzyme to break the cellulose. And then the cell walls becomes loosened, and the water enters the vacuole, and the cell elongates on the side where it is. You can also talk a little bit more about the water causing a higher trigger pressure in the vacuole, and that means stretching the entire plant. Now let's talk about micropropagation in plants using tissues from shoot apex. Now, if you're using tissues from shoot apex, then obviously you're talking about using the stem cells on the tip of the plant, and then place them on nutrient agar gels and growth hormones, and then causing new productions of plants. So micropropagation is a technique used to produce large numbers of identical plants or clones from a selected stock. So remember how you can use stem cells to make new clones? Well, it's the same thing here. You can use the stem cells of a plant to make extra plants that look exactly like the same. So when cutting a plant, right, it's used to reproduce asexually, and the native environment is called vegetative propagation. So that means you're just cloning. That's what it means by asexually. It's just mitosis, cloning, the exactly same thing. Now, the plant tissues are cultured in a laboratory in order to reproduce asexually. It's called micropropagation. So this whole process of micropropagation is just to get plant stem cells and clone them so you can make more plants. So how does it work, the steps? A specific plant tissue you want to work with. That means choose the plant you want to work with. And of course, it can be from different types of plants. You're going to sterilize them. The tissue sample, which is known as the X plant, is grown in a sterilized nutrient agar gel. The X plant is treated with a growth hormone, like for example Auxin, to stimulate the shoot and root development. That means you're putting a hormone to make these cells basically grow as a plant. And then the growing shoots can be continuously divided and separated to form new samples. That means remember, you can get stem cells from the tip of the root and the stem cells from the tip of the shoot, right? So when you grow one, you can cut again and grow another one, and can you cut again and grow another one? So imagine you cutting so many different roots and shoots, right? And taking their stem cells and producing so many clones. So now you're making 100 plants that look exactly the same. Once that the shoots are developed, the clone plant can be transferred to the soil. And obviously, when you grow these in a petri dish, in a nutri-agar, or in a beaker, then you can put in the soil and have many of that. So this is the process. You select the plant you want, then you stimulate the shoot. How do you stimulate the shoot? You give a hormone. That hormone will make the shoot grow. So you can see that it grows, right? And it grows by multiplying. Then what do? You can actually cut them. Also the same thing with the root. You can stimulate by every hormone, and you can see also have the root growing. So also the same thing. Then once you have so many of these productions, so many shoots and roots product, you can transfer that to the soil, and then voila, you have a new plant growth. So that's what the micropropagation steps looks like. Now, why would you use micropropagation used for? First, rapid bulking. When I say bulking, that means I'm making large amount of samples of it. So producing large new plants that are genetically identical. You just want to make a massive amount of exactly the same thing. You can also use for virus-free strains. What does it mean? It's propagating plants from the non-infected meristems. So that means sometimes plants can be affected by virus, right? And you want to find one of those plants who was not affected by the virus, and you want to make a massive amount of just that plant that was not attacked. And once you take the stem cells of that plant that was not attacked and make a massive amount of production of it, then you're going to have a bunch of plants who are very much free from that virus. And then, of course, propagation of rare species. Let's say one species of plant is dying. What you can do is take the stem cell of that species and just increase their numbers again, and they don't become species that are rare anymore. By increasing number of species that are difficult to breed sexually, increase the numbers of plant species that are commercially in demand. So one, you want to save the species that are dying. Second, maybe you want to have a large production of plants because you need this for the supermarket, or because some species cannot reproduce naturally. So in this right here, what you have is the reasons why you want to do micropropagation. So these are the three reasons. One, make a large amount of plants. Two, create just the plants free of virus. Three, you want to save some plants from dying as a rare species, or you want to make an extra productions for pharmacy or whatever it is. So these are the reasons why micropropagation will be helpful, which is basically it's the cloning of the plants. And that's pretty much it for plant growth, which is 9.3. Here is the study guide questions. This video basically answer all these questions that you need, and that's it. If you know these questions, how to answer them, then you have a very strong base on 9.3.